Welcome to another episode of the Bioinformatics Lab podcast. I'm Kevin Libwood with Andrew Page. We're both from Pathogen, and I forgot exactly what our intro is because it's been a little bit, but I think that's clear enough of an intro. Diving into the topics, I'm calling from Cape Town, South Africa, here for the first annual PHA4GE conference. So I think we have a lot to talk about in at least two episodes. In this first one, we'll at least describe what PHA4GE is, maybe talk a little bit about both yours and I's experience in the different working groups and where it's gone over the past 4 years and what maybe even the future looks like. And then in the second half of this, we can talk, or at least I can give a perspective of what's going on in the conference and kind of the big moves and announcements that have been taking place over the past couple of days. But even before we start, maybe we can start with the introduction of what PHA4GE is from your side of things, Andrew. What is PHA4GE and how did you start getting involved in things?
Well, it's the Public Health Alliance for Genomic Epidemiology, and there's the number 4 in the middle to match GA4GH, which is the human equivalent. And it's just a collection of people worldwide who have a real big interest and passion about public health. And it was decided a few years ago, hey, let's all join up and push in the same direction because pathogens know no borders. There's no point in reinventing the wheel 20 times. And so they got together a collection of public health scientists from around the world and academics, and the Gates Foundation put in some seed money to get the secretariat started up. And the first time I heard of it was probably 2019, maybe 2018. I can't remember. It was before the pandemic, and it spun out of, I think, frustration from GMI not actually having many outputs at the time, and people want practical outputs. So, like, say, for example, I'm in a working group and we look at data standards, actual usable things we can do, like Python libraries or specifications, physical things, not software, actual outputs that can be of use, immediate use to public health. What about yourself?
I got word of it also around 2018, 2019. I remember Duncan McCannell, who is at the Office of Advanced Molecular Detection, currently the director of that program, made that announcement at the US. I think it was maybe Advanced Molecular Detection Day over there at the CDC, and he talked about this consortium. And at that time, I wasn't really privy to a lot of the international goings-ons about things because I was really focused in my role in Virginia. But it was obviously a recognized need of collaboration and standards across the field and all the like. And I was a pretty active participant in the US side of things, but I was pumped to see that this organization was started out and had so much backing also from the CDC, because I had always wanted to be a part of the international community of practitioners. And it's also worth noting that Gates funded seed funding from Gates, a lot of CDC, US CDC involvement and leadership, but then really kind of headquarters, maybe you could say, out here in South Africa. So the principal investigator is Alan Christoffels, who is the director of the South African National Bioinformatics Institute, what I just call SANBI, out here. Well, hence, why I'm out here, in that, the first conference was out here in Cape Town. And there's obviously a lot of overlapping efforts from a lot of different groups. We've talked about Staff B in the past. We've talked about GMI in the past as well. And with PHA4GE, they was that focus of trying to get practical outputs as quickly as possible. And they approached it by dividing it into at least 5 different working groups that I can at least think of at the top of my head. You already mentioned the one you're involved in, from data structures. There's the workforce development working group, data infrastructure. I know there's ethics and data sharing. And then my involvement came by way of pipelines and visualizations. And it was 2018, 2019 was really timely for things to start, because we saw the need for international collaboration in full-blown, critical spotlight in 2020 during the pandemic. And that's when I really first started doing things, because I was first just invited as a participant to Pipelines and Visualizations work group, given my involvement in Virginia, as I said. And I think it was my working group was originally chaired by Torsten Seaman of MDU out there in Melbourne group. I forget who exactly else, someone quite a bit more senior than I was at the time. But then Torsten and whomever else that was got pulled to their main institutional duties, given their role in, at MDU and otherwise. So there was sort of a vacuum of no one was chairing our working group. And then I got tagged, I by Danny Park, nominated me to chair that working group. So I felt like I was a bit of the imposter syndrome of like, oh, I'm just this bioinformatics scientist from Virginia. I don't know if I can, make value in this global conversation. And the other chair on my end was Jamie Southgate, who had very little bioinformatics experience, but he was a secretariat of PHA4GE. So that was my first introduction of like, okay, let me first exposure to the international community and then quickly elevate it to chair. So over my time has been trying to figure out how do I make use of this working group. And I've been now a chair for 2.5 years. And I have a ton to talk about in terms of what I've learned, what we've generated and outputs. My now role is in how it's changed in PHA4GE. But yeah, it was definitely a cold splash of water into making international consortiums work for the pipelines of
So in my
visualizations.
working group, it's a data structures working group, and Emma Griffiths was, I think, deputy chair, but she took over as chair very quickly because people have different responsibilities, whatever.
Yeah.
And actually, I suggested the very first project they work on, which is now known as Harmonizer.
Nice.
So originally, I just before the pandemic, I'd created some software called Skigare, which is an Irish word, and it was for filtering AMR, right? And as part of
wait,
that,
Irish for what? Irish for what? I didn't need a translation.
Oh, I don't know. It might be filter. I can't remember. Obscure Irish words that is okay for Google,
Yeah.
But as part of that, I'd written some very pretty Python for parsing the outputs of lots of different AMR programs and putting them into a single, construct. And it's like, well, actually, this would be really good for BioPython, some kind of single. Really, you didn't matter what AMR program you used, you could just kind of slot them in and then you could use them and it would be quicker to write scripts. And this is exactly, what this kind of working group would find very useful. And so very quickly, the group was like, yeah, this is awesome. And so they pulled together, some software, hobbled it together, built, a whole system around it. And that's a very useful output to be able to take any output from an AMR program, because they're all totally different, and have more unified outputs at the other end. Just it makes scripting easier. And then the second things we went along and did were things like, I think SARS-CoV-2 came along at that point, and obviously the big thing was, well, there's no universal metadata standards, contextual metadata standards for SARS-CoV-2. There were some generic virus ones and pathogen checklists and things like that, but we really needed something drilled down, because you could tell even at that time SARS-CoV-2 was going to be like the pathogen that would be sequenced more than anything else in the world, and it's been sequenced tens of millions of times at this point. And so that's what they got out. They got out a drilled down practical specification. This is what it looks like in NCPI, this is EBI, this is what it looks like in GISAID, and really set out this is what we should be doing so the whole world can move in the same direction. And it got adopted broadly internationally, which is really cool, and it's a real key output, I think, of that committee, of that working group, to actually direct the biggest challenge of our time.
Absolutely. And even on our side with PHA4GE, we were working with US public health labs, navigating all the different databases from GISAID to SRA to Biosample and GenBank and all the like is quite difficult when there's not a map in front of you. So I think the working group helped to lay out that map and define what is critical, what is recommended, how to actually get this data into which database and all the like. So it provided a guidance, a template, a scaffolding for laboratories to quickly submit the most critical information about the samples that they're collecting and sequencing. And then you mentioned Emma Griffith, the chair, maybe co-chaired with also Finn McGuire, and was there another one before that, or?
I think David Arneson was a chair at one point.
Oh, okay, yeah.
But yeah, Finn was in effect, chair, deputy chair.
Yes, okay. And worth noting, both Emma and Finn and data structures being just a crazy prolific working group across the PHA4GE consortium, because if you want to say something on that, it's just amazing how many, like you said, practical.
I feel like fraud, because before the pandemic, I did attend all the meetings, and then pandemic hit, and obviously, I was redeployed to work on that stuff, so I dropped off pretty quick. But I've kept, up with some of the stuff they do, and it is fantastic. And one of the reasons is just having a good, strong chair with a set, tempo. So every 2 weeks I'd have a meeting, like clockwork, and it was very, not talking for the sake of talking, but actually getting stuff done. And that's the difference. You know, some committees are very technical focused, and it's just like, yeah, we know the problem, we're just going to work towards that, and then it's just about herding people in the same direction. And actually, what I love about some of the papers that have come out from this working group is, you know, there'll be authors from like 20 different institutions spread out across the world. You don't get that on papers like that. That's a rarity, you know. This is not just at the university saying, oh yeah, we've made up a standard. It's like people from all the major institutions and labs around the world saying, actually, no, we've gotten together, we've discussed it, and this is how we're going to do it. And so that means there's immediate acceptance and immediate weight to the value of it.
I think that's something Emma has done incredibly well. In given how prolific she's been and how that working group specifically has kind of set the tone for a lot of how the other working groups have acted. Definitely the one that I chair. In being community driven, getting the feedback as quickly as possible, getting an international perspective in all the different initiatives. Yeah, it gives us a precedent on how to act and what is effective for the works that we're trying to do. I would even go way even beyond that. I have just immense gratitude for Emma's perspective and what she helped me to do on our working group. Because when I first joined as a chair, as I mentioned, I felt a bit, out of place in being able to lead those kinds of meetings. But I got a one-on-one meeting with Emma, actually, I guess one-on-two with Jamie Southgate and I. We spoke to Emma and we were looking at what your guys' working group was doing. We said, how the heck do you do this? How do you have a group of all volunteers, make it effective, hold people accountable, and kind of keep driving the ship forward? And she gave us some really just practical advice, like what you mentioned, just almost religiously meet 2 weeks, even if there's no updates, just make sure you're meeting so you can have that continuity of conversation going on. And then putting out on paper what the plan is, how you're going to act, and what you want to do. And those things that seem so simple, it was really nice to get that from Emma and her perspective, because we employed that to the T on the pipelines and visualizations working group. And so when I came in terms of the products and focus that we were looking at was primarily for the past 2 years, SARS-CoV-2. And the way we utilized our working group, we had a group of technical practitioners from around the world, and we started with just meeting every 2 weeks. Let's all discuss for 2 weeks what's happening, what's going on in your lab. And then there's kind of a natural progression of what started happening every week is, Richard Neer was also in the working group, Danny Park and myself and a bunch of other, Peter Van Houston, of course. And we were all discussing the same challenges. It was like, oh, okay, that's a challenge you're working with, either at your lab or collaborators' lab. And then so what we just started to do with SARS-CoV-2 is, okay, let's define what are the critical challenges. And it was even something as simple as, okay, the first challenge laboratories are going to face is once you generate amplicon data for SARS-CoV-2 genomes, how do you generate a consensus assembly? And then from there, how do you call variants? Or rather, before that, maybe how do you trim the primers? And then how do you call the variants? And then how do you ensure there's not reference inclusion? And then how do you do pangolineage? All these things. It was really just what are the major challenges? And then from that, we can kind of look at that all as a group and think, what are the current solutions to address each of those challenges? And then we put that out. And then what kind of naturally emerged was a guidance document where we can hand to laboratories that were starting up the same kind of initiative. They didn't have to start from scratch. They didn't have to kind of dig the literature and, weave their ways into technical forums and communities. They could look at this guidance document and know, oh, okay, the first thing I'm going to need to do is ensure I can do consensus assembly, primer trimming, variant calling, etc. And they can go off and running. And we found this really valuable. We would distribute this. We made it available on GitHub Markdown. And then we allowed it to be sort of a living document. And so we did that for a number of different things during the SARS-CoV-2 pandemic. For example, when the Omicron variant first emerged, we quickly got the heads around in our technical community. What is the Omicron variant? Why is it important? The oddities of the genomic relatedness, the potential impact on public health. Let's get that all documented down and then put out another guidance document. When recombinant lineages were top of mind for a lot of laboratories, let's define exactly how you define a recombinant lineage, how you validate it and assess it and all the like. And so we kept that model. And then now what we started to do is use that same approach for other pathogens of public health concern. Now we have a working group, for example, that's focused on influenza. We have one focused on TB. We have one focused on enterics and all these other pathogens where they're as specialists in those specific pathogens. They're just defining what is the critical challenges and what are the solutions available. It's almost like a mini lit review. And it's created a little system of quickly getting these kinds of resources out there to the field.
Yeah, that's awesome. And like we're only on 2 of the different working groups, so there's lots of other ones, and
Yes.
they,
Yes.
obviously look at totally different areas, which is really kind of cool. Like phage is a really, really valuable resource there for the world, and hopefully it'll continue for many years to come. But I think that's
Yes.
all we have time for now, and we'll maybe come back next week and we'll discuss the very first phage conference.
Absolutely. And for anyone listening to this, if they're interested in learning more, we can turn them just to the phage website. It's PHA4GE. I think they have the .com. Am I right on that? Let me just make sure.
.org.
Oh, .org. Thank you. All right. And then they can join either our working groups or the others from there. And we'll get to the next episode.